Biologist. I don't know if anyone's on there just yet. I was having issues on the other one. Hopefully you guys will find this new link. I hope. Let's just see if I can go back, get some people on. I'm not very good. Right, hang on. Share that. Copy. Ah, hello, someone's made it. As you can see, I'm terrible with computers, so I've had to resort to using my phone. Oh, fantastic. You guys have found the new link. Ace. Great. Oh, fantastic. This is a bit of a surreal experience for me, really. I'm very, very new to this, hence not being able to use a computer to obviously start a YouTube live. So hello, nice to see you, nice to be with you this evening. So I can see, I apologize if I say your name wrong, Marielina is here. So hello, it's nice to see you. So from the comments that you guys have been saying, I understand that you guys want to go through what autosomal linkage is. And I put a couple of suggestions on there as well. Hello, SX Laninki as well. Nice to hear you and have you here with us. Hello, Chris. So good to see you guys. I have no idea how this evening's going to go. As I said, I'm very new to this. This is the first one that I've done. So please carry on asking questions. Hello, Labiba as well. So I'm going to go through autosomal linkage. I was going to go through some biological molecules, nucleic acids, classification, evolution, and also some biodiversity. But I'm very much open to suggestions from what you guys want to do. Hello, everybody. It's really good to see you here. So any suggestions for what you would like to go through first on any of that content? And I will start explaining as best I can. I did try to log on via the computer, but that just was not happening because I wanted to share some PowerPoints, but hey ho, we're here now. So, shall I start off with autosomal linkage, or do you guys have any other preferences? So I've had a comment there about biological molecules. More than happy to start off with that. Oh, that's fantastic. Oh, hi, Clumsy. Fantastic, that's really good news. So Clumsy joined my channel, last year and helped her all the way through, and fantastic. Well done for doing that marine biology. Okay, so let's have a look. Yes, get some revision done, she says, or he. Nice for that advice, thank you. So let's have a look at biological molecules. Now I'm going to be working through the OCR specification. I'm not too sure what specification you guys are on. If you want to drop a bit of a comment on there. I think the most popular ones are AQA and OCR. Now there is an awful lot of overlap between the exam board that I'm currently teaching and the other ones as well. So I think most of you do an OCRA, which is fantastic. So a lot of the stuff I'm doing here will be very relevant to what you're doing. I'm hoping there'll be a recording of this, Sophie, but I'm not very good with computers. So if there's the opportunity, I'll try and save that for later for you to watch. So on the OCR specification, I don't know if you have any notes available to hand, or if you have any space to make any notes, but I'm just going to run through the spec points dead quick. So the first spec point on biological molecules is hydrogen bonding in water. So hydrogen bonding in water, you normally get 3 marks for this. It's normally a 3-marker question. And again, if you want to try this along with me, so you can draw it as well if you have a pen and paper to hand, or if you have a whiteboard, you can draw at the same time. And I'll draw it as well, just so I can show you where the marking points are. So hydrogen bonding in water, if you want to give that a quick go. So H2O, if you want to have a go at hydrogen bonding in water, I'll draw it out as well, and then we'll go from there. So hopefully you've done something a little bit like this, I hope. So where we've got our 2 water molecules of H2O, and then we get our marking points here. 1 marking point for drawing a dipole on each of our hydrogens and oxygens. So that means that you're drawing a delta sign for slightly. So we use this term here, this slightly, and then positive. So slightly positive hydrogens, so that should be on all, and then slightly negative oxygen is what we should be having there. So we should do that. So you get 1 mark for your dipoles on them all, and then we get another mark for showing your hydrogen bonding and water. So in this case, I've done it as a bit of a dashed line, but you guys can get marks for a dashed line like that or like that, but definitely not a solid line like this one, not a solid line like this, because it's, yeah, it shows that it's a covalent bond rather. The other thing we've got to do is label it as a hydrogen bond. And then that is all your marking points. So those are your 3 marking points there. Those are your 3 marking points. So hopefully you guys managed to do something similar to that. So you normally get 3 marking points for a drawing of a biological molecule. The other thing that we need to talk about here are the roles and the properties of water. So the first thing is it's really helpful to link it into organisms here. The first one we'll do is the solvent one. So water is a really good solvent, which means we can dissolve substances in it. Now this is ideal for plants and animals. For example, we have a lot of water in our bloodstream, which makes it ideal for carrying substances around our bodies, such as amino acids or glucose or ions and minerals within the blood. Transport medium kind of links into that as well, where you have a lot of you can transport substances around the body. We've also got the coolant. Now the things that help with this one are the high latent heat of vaporization and the high specific heat capacity as well. So those 2 things help with water being able to access the coolant. And the reason is that because it takes a lot of energy to heat up the water by 1 degree to make it evaporate. So therefore it takes a lot of heat energy with it. So therefore that makes it a really good coolant. If you want me to repeat anything or go into anything in a bit more detail, please just drop a comment, or if there's any particular area of biological molecules you want me to go through again, just drop a comment in this section as well. So the other thing that makes water a really ideal habitat relating into that high specific heat capacity and the high latent heat vaporization is because water does require a lot of energy to heat it up by a degree. This means that I need a lot of energy to do that, so it stays at a relatively stable internal, a stable environment. And because of that stable environment, it means that the organisms that live in the water, the aquatic animals, it means that they can then regulate their enzymes and metabolic reactions within their body quite easily. So it's really good for the aquatic animals. We've also got ice acting as an insulating barrier here as well. So ice floats on water because it's less dense. Due to, I don't know if you guys do chemistry as well, but due to that lattice structure in the ice. So that's spec point A. Spec point E is basically looking up monomers and polymers. Yeah, I mean, I can skip ahead to fibrous and globular proteins, absolutely no problem at all. We can do that one. I'll just finish this little bit off on monomers and polymers. So we add on monomers to each other using a condensation reaction and removing water, and if I wanted to break them up, I add in the water in a hydrolysis reaction. So we can skip ahead to globular and fibrous proteins. Justine has just asked for that one, so by all means we can skip ahead to that one. So globular and fibrous proteins, the fibrous proteins, these are the ones that let me just go, sorry. Globular are 3D globular in shape, and these guys are normally used for metabolic reactions. So this is where you get your hormones and your enzymes. So your hormones and your enzymes, these are normally globular proteins. They're normally like functional roles. So for example, obviously your enzymes involved in metabolic reactions and your hormones in cell communication to trigger responses. Now, because of the structure of the globular proteins, because they are that 3D globular shape, it means that they normally have that tertiary structure. And because of that tertiary structure, it also allows them these globular 3D proteins. It means that they're soluble in water. Because we need them to be transported around the blood. Yes, we can have a look at ecology as well if you want to. I think it depends where you guys are up to, because with ecology as well, that's at the end of the year 13, which I imagine quite a few of you haven't covered yet, but just let me know what you want to do. So just going back to that globular protein. So in order to make that globular protein soluble in water, it has to have a lot of amino acids with the hydrophobic R groups in the centre. Because you've got those hydrophobic R groups in the centre and hydrophilic R groups on the outside, it makes it more soluble in the water or the bloodstream or whatever it is it's in. So that's the globular proteins. Now there are a handful of globular proteins that you need to be aware of on the OCR specification. One of them is that it's insulin, and then you look at the role of insulin when you get onto hormones in upper 6. And what you need to know about it at this stage is it has a fixed shape. We've also got hemoglobin, which is a conjugated protein. And this one, what we need to know for this particular part is that it can change shape. We've also got catalase, which is an enzyme, another conjugated protein, and this one has a fixed shape. So thanks for your comments on what else you want to go through. I will try my best to cover as much as we can during this time. So after globular and fibrous, we'll have a look at ecology, and then we can look at membranes as protein synthesis as well. So that's all we need to know at the moment on globular proteins. Let's go and have a look at the fibrous proteins. So in the fibrous proteins, these are the ones that provide structural roles within the body. So for example, your hair, your nails, yeah, sorry, just reading that comment there, it distracted me. That's a really good comment, thanks for that. The face behind the videos. I need to do more of these really, depending on how successful this one is. Anyway, the fibrous proteins, these are insoluble proteins, and this is because they have lots of amino acids in there with hydrophobic R groups. And the hydrophobic R groups, because they're repelling water, it makes them insoluble, these fibrous proteins. They have very, very strong tough ridges. So these fibrous proteins, these are the ones that have straight chained molecules, and they normally have a lot of hydrogen bonds between them. Now, hydrogen bonds in their own are very weak. However, because there are so many of them in these fibrous proteins, it means that overall it's a very strong molecule. So for example, the ones in your nails, the fibrous proteins in your nails have more hydrogen bonds and disulfide bridges in there in comparison to the one with your hair, which isn't as strong. Now, there are 3 main proteins you need to know about here. So the first one is keratin, which is in your hair and nails. And these guys have disulfide bridges in it for strength. Like I said, the ones in your nails have more disulfide bridges than the ones in your hair. Elastins, such as in the alveoli, in the wall of the alveoli, these ones are the ones that recoil to go back to the original shape, and it helps the alveoli to recoil. And the last one is the collagen, which is in the skin. It allows it to flexible, but you can't stretch it too much. And collagen is what goes as you get older, unfortunately, as you can probably see from a handful of my wrinkles, which means wisdom, I believe. All right, so we've gone through the fibrous and the globular proteins. I think the other person, Jessica, you mentioned ecology. Could you tell me which part of ecology you wanted to go through there? Because I'm not sure everyone's up to ecology and has gone through it all. But whilst I'm just waiting for Jessica to comment again, we can have a look at diffusion and osmosis across membranes. Amna, you've made a comment about this, about diffusion and osmosis. Is it the practical side you wanted to go through, or the theory side you want to go through? Just to clarify that. Oh, hello from France. Hello. You studying A-level biology in France? That's fantastic. Okay, so diffusion and osmosis. I'll start off with a bit of the theory. Yeah, no worries. Okay, so let's go through the theory of diffusion and osmosis. Okay, so yeah, just answering that question there on the globular and fibrous proteins. So the globular proteins, these have hydrophobic R groups in the center of the molecule, which help and hydrophilic on the outside. Whereas because the fibrous are all straight chained, pretty much all of it is exposed to the outside. It's because it's not in a ball. And with the globular proteins, with the fibrous proteins, they do have those hydrophobic R groups, but because it's not in a ball and they're not on the inside, because they're on the outside of those chains, it helps to make that insoluble. So there we go. Someone else commenting there, thank God I finished my A-level biology. It is hard, but with a lot of effort you can get through it. You can do this, guys. Okay, so I'll just go through the diffusion and osmosis theory, and then I'll answer a couple more questions. So with the diffusion can occur across a membrane. It goes from a high to a low concentration. Now, molecules that can diffuse straight through a membrane are small and non-polar molecules. So for this, it's things like your gases such as your carbon dioxide, your oxygen. So those are your small non-polar molecules. You can also get the only difference to this rule is water. Water is polar, it's the only polar molecule that can diffuse straight through the phospholipid bilayer. Anything else that is large or has a charge, such as an ion, like sodium ions or potassium ions, or anything large like glucose or amino acids, they can still diffuse through a membrane, but they have to use a channel protein to do that. They cannot just go straight through the phospholipid bilayer. They have to use a channel protein to do that, and that is called facilitated diffusion. Now, diffusion through the membrane can then also cause differences in concentrations as a result of those molecules moving. So just bear that in mind when you get onto exam questions on this. In terms of osmosis, at A level, we need to use the terms water potential here. So the osmosis is where we get a net movement of water from a high water potential to a lower water potential, down the water potential gradient by osmosis. And whenever you're talking about osmosis and what's moving where, there's like a couple of things that you should always say. And you need to use comparable statements. So you need to be saying, where is the water potential high? Where is it low? So by saying comments like the water potential outside the cell is lower than inside. So you've got to really make sure you're using comparable statements, not just saying low, like lower or higher than. Then you need to say that where the water's moving, so it's going into the cell or out of the cell, so wherever it might be moving in terms of that water potential gradient. And then we've got I've lost track of thought then, sorry. So the last bit is down the water potential gradient, and the last thing is by osmosis. Now, depending upon what the cell is, where the water's moving into or out to, it can have different impacts upon that cell. So, for example, if it is a plant cell, if water is moving into that plant cell, the plant cell will become turgid because the vacuole will fill with water and push against that cell wall. You've also got, if the water's moving out of the plant cell, it becomes plasmolysed because the cell membrane will pull away from the cell wall. We've also got in an animal cell, such like a red blood cell, if water's moving into my animal cell, it could eventually burst because it has no cell wall to stop it from bursting. If water was to leave my red blood cell, it would shrivel up. So hopefully that helps you a little bit with the theory of osmosis. So I've had a couple more questions on here. So I've got mocks in less than a week and I don't know how to go over content, any time suggestions. So yes, some ideas here on how to revise and cover content for mocks. A really good way to do is, first of all, identify your weakest areas and work on those. So you can do that by having a look at the specification, identify your weak areas, look at the Clare Biology, and you'll find that it's broken down into topics. So you can just focus on that topic and do a bit of blurting. So for example, watch a video on, I don't know, like osmosis, for example, and blurt it out, write as much as you can remember, then watch it again and just check, have you got all those bits and pieces in there? Another good tool to use is, as someone said on here, is the physics and maths tutor. So yes, you can use physics and maths tutor for end of topic tests and it'll help you for your actual A-levels. But the problem that I'm finding here with some of my students is they're using physics and maths tutor and then they're learning the mark schemes, and that's really you're shooting yourself in the foot by doing that. You really need to make sure you understand the content, which is why I'd focus a little bit more on understanding the content first and then assessing your knowledge, using things like physics and maths tutor or Seneca to assess your knowledge. But a really good way to learn the content and get stuck in your head is watching the videos on blurting, or if you have time, making flashcards or mind maps. Or another really, really good way to do it is working with someone else and asking them to test you and you to teach them, and vice versa, because that will help both of you guys to improve more. So hopefully that answers those questions there. What else have we got? It's a bit confused on CO dilutions. I think the whole world gets confused on CO dilutions. So, I mean, we can try and go through that as well. We've had a request to go through succession. Miss, you are the best, thank you. Also clear the difference between hyper, hypo and isotonic solutions. Yeah, so I'll clarify that because we have just done osmosis, and then we'll have a look at sieve tube elements as well. Okay, so hyper and hypotonic solution. So if you think of someone whose class is maybe hyper, you've got a lot of energy. You can move around, you've got a lot of energy. So if you're hyper, it means you've got a lot of something. So in terms of hypertonic, it means there's a lot of sugar within that solution. If it's a hypotonic solution, it doesn't have a lot of sugar in that solution. So that's what the hypo and hyper means. If it's isotonic, it means that the inside of the cell has the same water potential as whatever it's surrounded by, so the solution it's in. And that's what isotonic means. So if you're in an isotonic solution, there'll probably be no net movement of molecules, water molecules, across that membrane. You'll still get some movement of molecules. You can't say there won't be any movement, but you won't get a net movement, so a bunch of it moving in or a bunch of it moving out. So I teach OCR, scheme of learning, but it does overlap a lot with other content as well. So I think the other one was, so yeah, good question there about the phospholipid bilayer having a hydrophobic core. So how does water diffuse in? Because it is so small, because it is such a small molecule that acts in its favour here, which is why it can pass through. But because everything, and it's it is polar, but if you think about the structure, like for example, the hydrogen ions are only slightly positive, whereas if you have a full-on ion, which has a full-on ionic charge, because that is so polar, you won't be able to get through, but because water is so small and only has slight dipoles, it can squeeze through. But that is literally the only exception to the rule there. So we have a request to go over succession. So for this, I'll try and pull up a picture just to help with your understanding with this. So succession, this occurs, you need to learn this in the upper 6 content for ecology, which some of you might not be up to yet, but it looks like Jessica has covered this already. So I'll just pull up a picture on this and show you through it. If it will let me. Okay, so there are 2 types of succession. You've got primary and you've got secondary succession. So in a primary succession, this normally occurs where you have brand new land. You've got brand new land. This could be from a volcano that's erupted and some lava's come out and you've got this brand new rock that's not had anything on it. Now the first thing that happens here is this, first of all, the land is very hostile. And because the land is so hostile, it means that there isn't much around. It's a very harsh environment to grow anything in. However, you do get a pioneer species, and the pioneer species comes along and starts to grow in that particular area. Now, the pioneer species is really important because it helps to fix nitrogen from the atmosphere. It allows photosynthesis to take place. It can tolerate extreme conditions. And examples of this include things like mosses and lichens. Now, you've probably met a moss or a lichen before, and you probably have a good idea what it is. But these guys break down the rock to get what's known as a very thin layer of humus, which is a thin layer of soil. What we then do is we move on to an intermediate community. So what happens here is you get things like grasses growing, and they outcompete your pioneer species, which was the mosses or the lichens. Now these are also really important because they allow to sustain smaller invertebrates such as insects. So you have those opportunities there. So basically the intermediate community, so the grasses and small flowering plants, they outcompete your pioneer species. After that, you then get some more dominant plant species, for example, like your oak or your hickory trees. And then we get to what's known as our climax community, where we can get no further succession in this particular area. And this is where we have a more stable environment with less hostile conditions, and it'll be more biodiverse. You get a higher Simpson's index of diversity as well. So hopefully that helps with the succession side of it. What else have we got that you guys wanted to go over? We've done that one. Yes, sieve tube elements was one of the questions. What is a sieve tube element? Guys, if you have any other questions, please drop them in there as to what you want to go through. You're welcome, Jessica. Okay, so the sieve tube elements. So this is, we should hopefully know that the structure of the phloem consists of sieve tube elements and also companion cells. Now, the way that this works is because my sieve tube end plates, because the whole of the phloem is still alive, it means that it needs vitamins, it needs ATP, it still needs proteins, it still needs things to be able to survive. However, because of the structure of the phloem, the sieve tube end plates, which is this bit of the phloem, it has very few organelles. So it actually only has a tiny bit of cytoplasm, my sieve tube end plates. And this is where my assimilates are going to flow through in my phloem. Now, assimilates could either be your amino acids or your sucrose, and it can flow in either direction. And what happens here is my sieve tube end plates, they need what's known as a companion cell, which is like a bit of a friend, which will help it to make things that it needs. So this is what a sieve tube element is. This is the sieve tube end plate here at this end, which allows assimilates to pass through. Okay, so that is that question there on the sieve tube end plate. So we have a question about I'm assuming you need intra and intra-specific variation. Is that what you mean, Ali? I'm not too sure what your question is there. Is it intra and intra? Just let me know. So coenzymes and cofactors. I don't think there is an easy way to get that stuck in your brain. I think they're quite confusing, to be honest. And we can go over it, and I'll there's quite a nice little diagram that I can show you that will help you with this. Yeah, so we'll go through that as well, Ali, no worries about that. So one way in which I can remember is if you write the word in fact. So in fact, inorganic is the inorganic ions are the cofactors. That's one way I remember that it's made of inorganic. So the word, just remembering that word, in fact. In terms of what coenzymes actually do, that is where it gets a little bit more confusing. And you won't be the first one to be confused with this. I think, first of all, we need to remember that coenzymes are heavily involved with things like respiration and photosynthesis, which you'll learn a lot about when you get up to towards the end of upper six. So with the coenzymes, this does help to activate the enzyme, and it's not permanently bound to the enzyme. For me, a coenzyme acts a bit like a postman. It can take something from one place to another. They help to produce a certain specific shape of the active site, but they're not, and they do take part in the reaction, which is why coenzymes are so important when we get into upper six. In terms of cofactor, these ones are the ones that are permanently bound to the enzymes. I'll show you a picture of this one. Hopefully this will work. So I think this is probably the best image to remember for this one, where you have the cofactor that is permanently bound to the enzyme, but is further away, whereas the coenzyme helps to make the site, the active site. So unfortunately, I don't think there is an easy way to remember the two of them. I think it's just a case of spending that time to remember them. So the other question was intra and inter-specific variation. So intra-specific variation is variation between members of the same species, whereas inter-specific variation is variation of different species. So intra-specific variation is probably, for example, the differences between yourself and me. So we might have different coloured hair or skin, we might have different coloured eyes or blood type or whatever. That's intra-specific variation. Inter-specific variation may be that the difference between, for example, us as humans in comparison to dogs, different things there. So hopefully that answers your questions so far. If you have any others, please drop them in the chat. If you, I think someone mentioned protein synthesis, is that right? Does someone still want to go through protein synthesis, or is there any other topics in particular you guys want to go through? Hopefully you're enjoying a bit of FaceTime. It's a bit It'd be nice to hear you guys and see you guys as well, but unfortunately that's not how this works. Just on that, if you would be interested in like a group session with smaller groups, can you just drop in a message that says just group, just so I've got a bit more of an idea as to what you guys are thinking. Oh yeah, autosomal linkage, we'll do that one now. So autosomal linkage, autosomal linkage. So in autosomal linkage, what happens here is, so this is linked into patterns of inheritance, which obviously some guys have done here. This is an upper 6 content as well. So in autosomal linkage, what happens is we have, it's all about genes being inherited together or not being inherited together. So for this, you kind of need a basic understanding of meiosis as well. Because in meiosis, what would happen normally is during crossing over in prophase 1, the genetics of the homologous chromosomes would be mixed up. And what we're saying here is, in the end gametes as the end result, if 2 genes are close together on the chromosome, it's less likely that they'd be mixed up during prophase 1, during that crossing over, to result in a variation of gametes of that particular gene. I'll show you an example in a second. Whereas if I have a gene, for example, up here and a gene down there, it's more likely that they'll be changed, the genetics will be changed via crossing over, and therefore result in different gametes. But I'll show you an example of this in a better picture than I can describe. So here we have 2 homologous chromosome pair, you can see. And in this one, you can see I've had crossing over occurring. Now, because in this example, genes A and B are quite far away on the chromosome, crossing over has actually split up these, so that in the gametes, I've got different variations of each one. Whereas when I've got autosomal linkage, what happens here is genes that are very, very close together on the chromosome are very unlikely to be separated during crossing over. So autosomal linkage is basically where I have genes that are on the same chromosome and they're said to be more linked because they're more likely to be inherited together. So hopefully that helps to explain autosomal linkage. Now in an exam, you'll probably get, this is more likely to get linked in here in terms of explaining why a ratio isn't what it should be. So for example, sometimes you should get ratios of 3:1 or 9:3 to 3:1, but sometimes due to autosomal linkage, you might not get some of those ratios. So hopefully that answers your question on autosomal linkage, which was quite a popular question. Was there any other questions you guys wanted to ask? So Labiba is interested in doing a bit more of a small group where we can walk through exam questions. If anyone else is interested in stuff like that, please do just let me know and I'll see what I can do. You'll never be asked to draw an exam, to draw an inheritance cross for an autosomal linkage. The only thing you need to know about this is that it's more like you might get it in a multiple choice question, or you might get it as a result in a short answer question as an answer to something as to why something hasn't followed the pattern it should be. The only crosses that you're going to have to do are the ones that are in the spec point. So that is, just let me remind myself, a monogenetic inheritance, a dihybrid cross, multiple alleles, sex linkage and codominance. Those are the ones that you're going to be asked to draw out in a cross, which I do think is due this academic year because it wasn't on in much detail last year, if I'm being honest. In terms of the nitrogen and the carbon cycle in, I think again that's ecosystems, I think. Yes, you do need to know all the steps and you need to know all the organisms and the names of the organisms involved. And again here what they like to do here is they like to give you a diagram or a picture and they might leave some blanks in there. I've also seen it, I mean I don't know if you've noticed it, but at the end of your exam you always have a short paragraph of writing with bits missing. Words missing, and I have seen the nitrogen cycle in part of that. But I think the nitrogen cycle may have been asked about last year. I'd need to double check that. I need to make my predictions for this coming year, but they'll be released later on. So I don't think you need to worry too much about the nitrogen carbon cycle. But having said that, they could literally put anything on there, so I don't know. So hopefully that answers your questions there, Jessica. Were there any other questions that you wanted to ask me on any of the content in upper or lower six? I'm happy to go through a bit more of a topic. If not, we can call it an evening there, unless you guys want to go through anything else. It's been really nice speaking to you guys. Hopefully you've enjoyed it. Hopefully it's been helpful. Can do 1 or 2 more of these. When have you guys got your mocks? Have you already done them, or are you going to do them? Is there anything in particular, what your weak areas, or anything in particular you want to go through, or how to answer a certain question? I'm happy to take any questions, really. And if you guys don't have any more questions, I could go back to doing biological molecules. Just let me know what you guys fancy doing, and we can do those. Oh, Jess has got hers on Monday. Good luck. Good luck. Just remember to lay out your 6 markers in that certain way. Random questions on walkthrough sessions. Yeah, no worries. Oh, Nayab's done them. How did they go, Nayab? Hopefully they went well. Hopefully we didn't use the words it, they, amount and size. Hopefully. But well done, guys. Really well done with that. So one of my students is on here as well, which is good. How to get from a B to an A. Yep. 1 week to mocks and paper 2 on Monday. Oh, good luck. Good luck. So in order to push yourself up to those higher grades, I would say that in order to reach those higher questions, for me, it's about the suggest questions. And the suggest questions are hard. And I think you need to familiarise yourselves with things that are beyond the specification. Learning the content is good, don't get me wrong, it's good. But learning the content and describing or explaining is only going to get you so far. Like, you need to be able to become familiar with harder content. And the only way you're going to do that is really by extending knowledge beyond the spec. So, for example, really easy way to do it at the moment is doing stuff like watching Planet Earth. Planet Earth 3 is out at the moment on BBC iPlayer. Simple things like watching that and making links to your specification will really, really help. But also you need to think about links between topics. So for example, I think today I was teaching about plant hormones. This is a really common exam question, really, really common exam question. The reason why they really like this is because it links up several topics together. So you've got plant hormone responses, you've also got hormones it links in, you've got communicable diseases it links in, you've got things like communication and homeostasis, you've got biological molecules. So you really need to be thinking about links between your areas and your subjects, and I think that will help you get from a B to an A. But as well as doing that, just do as many practice questions as when you've learned the content, and do as many practice as you can. So good luck with that. Good luck with that. Some other comments. So some have got their mocks after Christmas, which is well, good luck with that, because you've got Christmas to revise as well. Yeah, so in terms of other websites and recommendations, some of the stuff on Seneca could be quite good as well. But I do think I think the best thing to do is to do past papers. So if you're an A2, for example, an upper 6 student, you can do any of the lower 6 content now. So you really should be practicing at this stage doing full papers in timed conditions. Some of you may be up to the point in upper 6 where you might even be able to do a paper 2 content by now and sit a whole paper 2 on your own. So I would advise doing those, and those can be accessed straight from the OCI website. Other things as well to look at are things like the OCR, they do exemplars. Yeah, the breadth and depth are lower 6 contents. You can do them on your own now. So look, yeah, OCR does exemplar questions, and it's really, really helpful to go through those and look at it and think, how many marks do you think it is, and then have a look how much the examiner gave, because some of them you might find might be quite harsh. So I mark for the exams in the summer, and students are always losing marks for doing typical things. So for example, some of the stuff that I try and advise you do is when you're doing a 6 marker, to lay it out in a way that's really, really easy for the examiner to mark. So for a 6 marker, it might be, for example, an evaluate question. If it's an evaluate, you always do a for, against, and validity, for example. So I would always put, I put subheadings in, or I'd physically split up your question. Because as an examiner, when you're marking the question and someone's done that, you're like, oh, what a beauty. What I can do is I just award it as many marking points as you possibly can, because it's obvious that they've split it up in a certain way. So yeah, the only other website I know we can get questions from is Seneca, and they do sometimes do stuff like A star or A bits and pieces. Yes, you can use a bullet point and a 6 marker. For OCR, you would never get a 9 marker. So if you have seen a 9 marker somewhere, that's probably because it's an old specification one and not a new one. So hopefully that answers that question, Nab. So Educast and WJC is slightly different. There are some things on there that aren't on the OCR spec. For example, I think you might need to study cancer in a bit more detail on that one, whereas OCR, we don't really need to do much on cancer. Yeah, so yeah, no 9 markers. So most of the content that we teach on OCR is very, very similar to the WJC board. It's just a handful of things here and there. I think there's a little bit in upper 6 and a little bit in lower 6, not loads. That is just not on the OCR or the other way. Thank you very much for that comment, Captain. Some great names here. You guys have got some fantastic YouTube names. Okay, so we've got about 20-ish minutes left if you guys want to ask any more questions. Was there any other content in particular you want to ask, or any more advice you want from an examiner, so myself, on how to answer questions or anything like that? I'll go through one more advice as well in a second. So one thing I advise that you do in your exam is, say for example you have got a table or a graph or anything, any question really. If you annotate your question or if you annotate your graph, you can quite often get marks for that. So for example, there was a question a few years ago about identifying on a graph where the primary and secondary response was. So you could physically write it in terms of, you know, your primary response is where you, after you've been infected, and your secondary response was when you were injected. You could describe it, but how much easier would it be to just draw on the graph, this is where it's primary and this is where it's secondary. So please just write on the graph, and you can actually you can draw on the graph and say to the examiner, see diagram above, or use diagrams in your answer to support what you're saying. So yes, you can use bullet points. Yes, you can use diagrams to support your answers. So for example, if you've got a question asking you to describe hydrogen bonding in water, don't use words, just draw it. So don't, make life easier for yourself and the examiner. So thank you for that comment, Princess Random. Got you from a U to an A, which is fantastic, so well done. So the common mistakes normally seen on a 4 or 2 marker is not actually answering the question. That is the typical thing that students do wrong. For example, it asks you to describe and someone has explained, or it asks you to use figures from the diagram or talk about the diagram and someone just waffled on about something else. So the differences between describe and explain, for example, if I was asked to describe a graph, I know it's not the greatest graph, but hopefully you get the idea. If I was asked to describe this graph of enzyme concentration, I would say, describing, I would say, as the concentration of enzyme, for example, concentration of enzymes increases, the rate of reaction increases up to, 50%, whatever that concentration was, and then it would decrease again back to 0. That's a description. If you're describing why, it'd be something like, as the concentration of enzymes increases, I get more successful collisions through my active site and my substrate, therefore rate of reaction increases. So it's much more about the description about that. Also, students just don't use information from the questions. So you need to use examples from graphs and tables in your answers with units. Like you've got to start doing that more, because literally the answer is in the question. The answer is in. If you've not been taught about it, then the answer is in the question, and you've just got to be pulling that back out. So someone's asking when the next live stream will be. I don't know. Depends on this. I think it's going all right. Hopefully you guys are benefiting from it. But I'm more than willing to do another one, maybe in a month-ish time. We'll see. Or maybe just before in January. In terms of your maths and biology, I actually have a recommendation for a book. Yeah, it's this one. This book is really, really good. I don't know if your college just sell it on. We normally buy these in a cost price and sell them on as cost price to students. I think if you were to buy it brand new, I think it's about £6, so it's still not extortionate, but it's just essential maths for A-level biology, because 10% of your marks in biology is maths. So the more you can work on your math skills, the higher your grade is going to be. So if you think about it, 10% of a grade in one exam paper is a grade and a half in one paper. And that's how close the grade boundaries are within one paper. So I highly advise that you splash out and spend £6 on a book that, I don't get sponsored by anyone. I'm not quite that famous yet, but if it is helpful, that you can do this. And in those books, it goes through every single part of maths that you need to do for A-level biology, and it explains it, then it shows you a worked example, then it gives you exam questions. So I think that is a really good way to improve on your maths in biology, because you can target certain areas that you're weakest as well. So in every single exam that you're going to do, you will get some maths. In every single year, you get a percentage change every year. In every year, you will get a statistic. So that is either Simpson's index, Spearman's rank, the t-test, Simpson's index diversity, or chi-squared. You will get one of the big five every year. And I think, and I need to work on this, but I think it might be chi-squared's turn. But again, I need to have a look and see which one. But you need to make sure you're familiar with all of those, because at least one will come up. Do we need to know what Q10 temp coefficient is? Yes, I believe you do. I don't think that is provided in your exam. I think you need to be able to do that. Weird way of wording maths questions. I do A-level maths and sometimes get them wrong. Oh dear. You picked biology to avoid maths, so that was a mistake. It's crammed full of maths. But we could do one of these we could do another live session just focusing on some of the maths content if you wanted to. We'll just see what you guys think and what you want to do. So do you have any more biology content questions before we close this session? Feel free to ask anything, or even a suggestion for next time. I'm not an expert at YouTube Live, as you can tell. I did try to go on through a streaming platform so I could show you some PowerPoints, but obviously that didn't work. So I'll do it like this next time, I think. But if you have any suggestions about what you want to do for next time, or any questions or queries, good question. Is it Ada or Ada? Is it recorded? I think so. Yeah, so we can focus on maths in biology for next time, because that is on every single year. We can focus on the big five, as I called them. You will get a statistical test for each one, or one of them at least. And they're always asked about, so we can go through that. Okay, so if there are no other comments or questions about the biology, then we can call it an evening. Unless you have. Yes, we can do excretion next time, and we've got a bit of time now. We can do a bit of excretion if you want to. If you want to be a little bit more specific about what part of excretion, then we can talk about that in a bit more detail, because excretion is quite a big topic. Oh, the loop of Henle, that beauty. Yes, we can look at the loop of Henle. Okay, loop of Henle, this is in upper 6. This is in excretion and selective reabsorption. Okay. So this is where sharing a PowerPoint will come into hand. Anyway, let's go with it. Okay, so we'll start off with selective reabsorption, and then we'll move on to the loop of Henle. So hopefully we're okay with ultrafiltration, which occurs in the glomerulus of the Bowman's capsule. Hopefully we're okay with that a little bit. After the Bowman's capsule, we get into selective reabsorption, which occurs within the proximal convoluted tubule. Now, in the proximal convoluted tubule, I find it easiest explaining this diagram, which I'm trying to pull up for you. I just find it easiest way to explain it in terms of what's going on in the different parts. So in selective reabsorption, the first thing that goes on, I'll just pull up this picture for you eventually. that's helpful. Okay, right, here it is. So in selective reabsorption, what we have, you can see me there. You don't want to see that. All right, we have, on this side, we have the bloodstream over here. And on this side is the lumen of the proximal convoluted tubule. So the first thing that happens, we're going to focus on this side, is that the sodium ions here are actively transported out of these cells that line the proximal convoluted tubule wall, and they go into the bloodstream, these sodium ions. Now, because sodium ions have been pumped out of here, they're going to be in a low concentration inside this cell. So therefore, sodium is going to come in to the cells that line the proximal convoluted tubule wall. And they're going to come in using a co-transporter protein, which is this, with a glucose or an amino acid. Now, because glucose or an amino acid is also moving in using this co-transporter protein, it means that water is also going to move in by osmosis. And then because I now have in this cell, I have the glucose or an amino acid, they then move out of this cell into the bloodstream via facilitated diffusion. So they're going from a high to a low concentration gradient to be reabsorbed back into the bloodstream. And as a result of glucose or an amino acid going in this direction, it also means that water will follow via osmosis because it will lower the water potential of wherever it is going. In terms of, so hopefully that answers your question on selective reabsorption. So the main thing here about the active transport is this bit here in terms of the sodium ions being actively transported out of these cells that line the proximal convoluted tubule wall into the bloodstream. So the last thing that we'll take a look at is the loop of Henle. So the loop of Henle, hopefully we recognise this image. So we have the cortex of the kidney and this part, and it reaches down into the medulla of the kidney. The first thing we'll look at here is the ascending limb, which is this part. Now, as you can see here, in the ascending limb, the sodium and the chloride ions are actively transported out of this area into the surrounding tissue. Now, don't forget this surrounding tissue is part of the medulla. And it's going to lower the water potential. Oh, my watch is going off. It'll lower the water potential of the surrounding tissue here in the medulla. Now, what happens here is this part of the loop of Henle is impermeable to water, which means no water can enter or join into the loop of Henle at this point. So therefore, water needs to move out of my descending limb via osmosis. So water will move out of the descending limb, but it would also move out of the collecting duct as well. Now, just going back to this side, now because water is leaving my descending limb via osmosis, because the water potential of the surrounding tissue is lower, it means that the substances inside of here, inside of the loop of Henle, means that I'm going to get a higher concentration of solutes inside here. Now, because of that, my sodium and chloride at this part of my loop of Henle will diffuse out into the surrounding tissue. Now, because of the movement of ions here by active transport and here by facilitated diffusion, it means that the water will move, it means the water potential will decrease as I go down the medulla, and therefore reabsorb more water. So if you've just joined, we're literally just finishing up, and that is the loop of Henle. So hopefully that helps. So the next one that people want to do is the maths one. Guys, you just give me a bit of an idea as to where we're up to. I think the majority of people are year 13. We've got someone who's asking a lot of questions, someone who's year 12. But yeah, hopefully that's helped you guys. I think one or two people are interested in a bit more of a one-on-one session. But other than that, guys, well done. Hopefully you've enjoyed this evening. I'm sorry about the bit of a mix-up logging on. I found that quite difficult. I'm not very good technical-wise. But other than that, I hope you've enjoyed it. I'll try and save this video if I can do. And good luck with your studies. Don't use the words it, they, amount and size. Hopefully that's helped. Been nice to meet you and speak with you all. Bye-bye.